One of the first things you learn about in hands-on microbiology is the fundamental method used to classify bacteria, the gram stain. By staining with crystal violet, rinsing, and then staining with safranin, bacteria are differentiated into two groups. Gram-positive if they retain the crystal violet, which makes them purple, and gram-negative, indicated by pink, if the rinsing step removes the crystal violet, allowing safranin's color to show through. This categorization does have a structural basis. Gram-positive and gram-negative bacteria do in fact have distinct cell envelope architectures that account for the differences in how they stain. We call gram-positive bacteria monoderms because they have a single cell membrane, which outside of it has a thick layer of peptidoglycan, the material that makes up the bacterial cell wall. In contrast, gram-negative bacteria are called diderms because they have, outside of a thinner peptidoglycan wall, a second cell membrane. That gram-negative cell membrane is a lipid bilayer, but it is not a phospholipid bilayer like we see in the membranes of most other cells. The inner half of the membrane does have phospholipids, but the outer half does not and is entirely composed of glycolipids, like LPS. My first assumption when I hear about a system for classifying organisms is that it reflects their phylogenetic ancestry. Does a monoderm-diderm split represent one of the most fundamental divergences in the bacteria, and by consequence life as a whole? The first question we would have to ask to answer this is whether monodermy or didermy is the ancestral state of bacteria in general. Where did they start? Despite the fact that the majority of bacterial phyla are diderms, I like the idea that the monoderm envelope is the ancestral state because it is structurally closer to archaeal envelopes, which I think kind of suggests that their last common ancestor in some way resembled a monoderm, structurally speaking. However, the question is still absolutely up for debate. And so if we go from the assumption that monoderms are the ancestral state, all of the diderm bacteria actually have similar enough envelopes to suggest that didermy arose a single time, out of the ancestral monoderm bacteria. What throws a wrench into this though, is that some of the phyla of monoderm bacteria, like the Firmicutes, also have diderm bacteria in them, and not even all of the monoderm bacteria within the Firmicutes form a monophyletic clade. There are two immediate explanations for this oddity. The first is horizontal gene transfer, i.e. that some Firmicutes that were formerly monoderms got the genes needed to become diderms from some other pre-existing diderm bacteria. However, this idea has been disputed on the basis that it would require the transfer of several different genes basically simultaneously, which one could argue would be implausible. And so the other idea would be that the Firmicutes are ancestrally diderms but repeatedly lost the outer membrane to become several times independently monoderms. However, our understanding of this is pretty rough because bacterial phylogenies tend to be poorly resolved. In conclusion, the gram stain is a proxy for whether a bacteria is a monoderm or a diderm. These classifications reflect fundamental differences in cell envelope architecture, but do not form monophyletic clades. It also bears noting that many bacteria give inconclusive results from the gram stain, and many don't fit nicely into gram-positive or gram-negative categories. I want to cover that in my next video. But as I've touched on, a lot of our challenges here come from a lack of resolution or certainty in how we determine bacterial phylogenies. The relationships between bacteria from species up to phyla are often not established with very much certainty. One could argue that this is because trying to classify bacteria into species at all is a bit of a false effort. The Linnaean taxonomic classification system was developed with plants and animals and maybe fungi in mind. And so to assume it gives us a good system for understanding the relationships between bacteria might not be entirely true. Bacteria reproduce and share genetic information in ways very different from multicellular organisms. For example, we lack the horizontal gene transfer capabilities of bacteria, with our genetic information strictly passed from parent to offspring vertically. Furthermore, while bacteria reproduce asexually through binary fission, the predominant mode of reproduction in multicellular organisms, especially animals, is sexual. Perhaps a better model for understanding the diversity of bacteria, rather than a tree of life, is something more akin to a net, with interleaved threads rather than perpetually diverging branches.